So Pseudomonas syringae is an important pathogen of many plant species, and cherry included. So if you can take a look at these pictures, syringae can infect and cause canker symptoms on different parts of cherry trees, and that results in the damage to trees and also loss of reduction. So Pseudomonas bacterial canker have been considered a very important problem for global cherry fruit industry. It's very difficult to control this disease, mainly because there are various pathogens of Pseudomonas syringae. It's a species complex, and also within each pathogen, there are different races of Pseudomonas syringae, which can target different host cultivars. So a number of Pseudomonas lineages have been found to cause bacterial cankers. However, interestingly, there are also other non-pathogenic bacterial lineages found in cherry trees, which have the potential to be pathogenic. And these bacteria are considered as epiphytes, which are widely spread in cherry trees in different orchards across the UK. It's very important for us to know more about the interaction between Pseudomonas syringae and cherry trees, especially how these pathogenic strains evolved and adapt to the potential host, and thereby we can come up with better idea for disease control and risk management. So about this whole project, there are 3 work packages. I'm working on the first work package, which focuses on the bacterial populations in cherry trees. And the second work package is about the factors which affect the host range. And this work package will be further explained by Mojgan. The talk will come later. And finally, with the findings obtained in the first 2 work packages, another postdoc at NIAB, Stasius, she's going to develop a tool for predicting host range of Pseudomonas syringae use machine learning methods. So the previous research found that there is an enormous diversity of potentially pathogenic and non-pathogenic Pseudomonas syringae on the leaf surfaces of cherry. And also there is a large regional variation in Pseudomonas populations. In addition, there is significant variations in the Pseudomonas populations in cultivated cherry and white cherry trees. So in my project, in the first work package, I'm going to look into the temporal and spatial variation in populations of Pseudomonas syringae by carrying out multi-site and multi-year survey of the Pseudomonas populations in the different orchards across the UK. And also, I'm going to compare the epiphytic populations of Pseudomonas between the cultivated cherry trees and white cherry trees. And finally, as agronomic practice could play a role in shaping Pseudomonas populations, I'm going to investigate whether the application of nitrogen or the use of polytunnel covers would affect the Pseudomonas syringae populations by doing field work. So I'd like to thank everyone who worked with me to help to improve the cherry security, the security of cherry production. And now Mojgan from University of Birmingham is going to tell you more about work package 2. I've stopped sharing.
Thank you. So hopefully you can see my slide and you can hear me. All right? Yes, perfect. So hello everyone, my name is Mojgan. I'm a postdoc working with Rob Jackson based at University of Birmingham. I'm working on work package 2 of cherry canker BBSRC funded project. And we are working on how new strains of pathogen emerge and evolve on trees. So as Tracy explained, bacterial canker is a major disease of Prunus species, including cherry and other stone fruits. and it is caused by Pseudomonas-Prunus species complex, syringae, and Monosporanorum. So I took these pictures during my last postdoc, previous postdoc, so I just wanted to share with you. So as you can see, the pathogen is able to infect throughout the year, infect the woody tissue that the wood produce huge amount of gummy ooze, and on leaves you can see the shot holes. So it's a black-brown spot that drop out and produce the shot hole symptoms on the leaves, and also cause the necrotic region on fruits. So annual problem for global cherry fruit industry, and has a huge economic loss. So currently, as Tracy said, there is no cure for this disease. So it is very important to understand the pathogen behavior and evolution through this research. So what we know about this disease, that there is significant variation in resistance to canker within cherry cultivars and also between genotype of wild cherries. Also, we know that there are epiphytic population, as Tracy said, living on the surface of non-diseased cherry leaves. And we know that these epiphytes might have genes that predicted to have a key role in host adaptation and pathogenicity to Prunus species. But what we don't know is whether this epiphytic population of Pseudomonas-Prunus have pathogenic potential. So previous research found that there were effector genes that appear to be convergently gained in cherry infecting lineage or also significantly lost. So it shows that this effector gain was due to either plasmid or phage-mediated transfer of effectors. So there are a lot of unknown about this disease. So what we want to find out during this work package, we want to understand whether the epiphytic population of Pseudomonas-Prunus contain effectors that we predict may alter epiphytic persistence on cherries. So we want to understand the effector that regulate epiphytic ability to persist on trees. So we will do like leaf inoculation, we will do effector gene knockout and deletion to understand the role of gene or genes of interest. We also want to understand the mechanism of host range expansion and host jump onto phylogenetically close or distantly related hosts. So we want to understand the genetic factor that may affect pathogenicity, virulence, or lead to new disease outbreak. So we hypothesize that following a host jump, it should be easier for co-speciation between host and pathogen to occur, or for further host jump to occur when hosts are phylogenetically closely related. So during this work package, we will do passaging, DNA extraction, and genome sequencing to see any SNP changes in the genome. So we want to understand if epiphytic population are able to evolve the ability to become pathogenic. So we will conduct the evolutionary experiment and passaging and genome sequencing to understand this role. And also we want to understand gene exchange by phage. So we know that bacteriophages are viruses that infect bacteria. They can incorporate their genetic materials into the genome of bacterium, and they become prophage. And then under special circumstances, they become activated. So whether an effector gene can be transferred via phage, and if this occurs in the cherry phylophylline, will be explored in this work package. So the preliminary work showed that there is the effector gene of AR1, for example, is found within a prophage in Pseudomonas-Prunus Monosporanorum, race 1, that predicted with high probability of being active. So we will purify the prophage first to check if it is active, and then we will check if the prophage containing the effectors can be transferred from one bacteria to another. So why we are doing this research and what are the impacts? So understanding the genetic mechanisms and effectors that are important in niche adaptation, and also understanding the risk epiphyte pose if they become pathogenic. So it will help us to develop precision diagnostic for Prunus species as well as in other crops. Also understanding how easy to do host jumps onto phylogenetically related hosts, will have impact for disease management, for example, choice of host to plant, as well as improved knowledge that a novel pathogen may or may not be better able to adapt to native eucalyptus species. So establishing if phagers are actively transferring virulent gene within bacterial population has implication in the use of this organism as biocontrol agent in terms of ensuring that no integration of genome can happen. So in overall, to summarize the impact of the whole work package in BBSRC funded projects, we know that the movement of disease through global trade is having significant impact across many crops. However, being able to trace the source of disease outbreaks and the origin is often challenging. So this project helped to provide more detailed information about whether imported plant material carry any potential pathogen affecting them and other close or distantly related crops. So it would be a powerful tool in shaping both rapid responses and also plant movement policy. So thank you all for listening and